Good afternoon, everyone. I would like to start by asking you a simple question. Has any one of you taken any form of plant-based protein this morning or in the afternoon?
It was yesterday morning.
All right. So then I would say we belong to a generation of people whereby we can say our source of protein is quite diverse. We've got quite a lot of what we eat as protein coming from plants instead of traditionally coming from animals. Okay. So my research is based on the issue of understanding plants, getting to know about plants, and then using the right tools which have been developed by system engineers to analyze plants and give us a better insight. And the whole philosophy or the whole motivation behind such thing is for us to be able to grow plants in a more sustainable way and to have more for less. Understanding stress in plants from response to changing light. We all, as living creatures or living things, have our various ways in which we behave whenever we have light. During the summer, we would like to have some outdoor activities because the light has a positive impact on us as it affects our metabolism. So the same thing happens with plants and animals. So particularly plants, they use light to manufacture their own food through a process called photosynthesis. So when you have photo, that's light, and then you have synthesis, like the process of producing food. So plants basically take in nutrients from the soil, and then through this process of photosynthesis, like the engine inside the plant, works very hard to produce food. So once it produces that food, then the plant can be able to use the food and then to sustain itself. So in the process of food production by plant, three things happen. Some part of the heat, some part of the light, which the plant has absorbed, gets emitted as heat. It's called non-photochemical quenching. That's the technical name, but you can just call it heat. Then another fraction of that light gets emitted by the plant, and that is called fluorescence. Fluorescence is a phenomenon whereby living organisms tend to emit light at some certain wavelength. So with fluorescence, you can be able to distinguish and know what exactly is happening with the plant. It's quite a diagnostic tool that has been used by plant scientists to know things about plants, such as deficiency in nutrients, and other things such as having problem with water or having problem with climatic conditions. So basically, you can say plant stress is divided into two kinds. We have what we call the biotic stress, and then we have what we call the abiotic stress. So biotic stress are stress caused by diseases, while abiotic stress are stress caused by other factors, such as the absence of nutrients or nutrients being depleted in the soil, or when you have some climatic factors that affect the production of plants. So for this, my research, we are narrowing down on a single kind of stress, and that stress is called phosphorus deficiency stress. Phosphorus is a very, very important element. At the moment, most of the phosphorus in the soil has been used up by farmers. So without phosphorus, it becomes very, very difficult to produce food. On the flip side, if you would like to depend on chemically produced phosphorus to grow plants, that would mean you would have to put in more amount of chemicals in producing food. And that is a number that you like the number to go down as much as possible. So what's the best way to go about solving this problem? The best way is to deploy the use of precision agriculture. And precision agriculture definitely has a lot of facets, among which we have the diagnostic part and then we have the production part. So the diagnostic part is you being able to use minimally invasive methods to be able to tell whether a plant is healthy or a plant is sick. So what we did in our lab was to grow three kinds of crops. So the three groups of crop of the same kind. So we had a susceptible wheat plant, and we grew the plant under the same climatic condition, such as light, humidity, and temperature. but we varied the amount of phosphorus which we use to grow the plant. So we had a plant which has the 100% required phosphorus, and then we have the second group having just 50% of the phosphorus needed, and we had the third group having no phosphorus at all. So these plants were imaged using a device which we've been developing in the lab, as well as the algorithm for the device. This device, what it does is it tries to replicate the behavior of sunlight. When sunlight gets into a plant, it produces food, it emits energy, and it also emits fluorescent light. So this device now gives in a light which is changing slowly with time. So when you send a light that is changing slowly with time into a plant, what this light does is it makes the plant to emit fluorescence, which gives a rather unique pattern. So this is the fluorescence that comes from a healthy plant, and then this is the fluorescence that comes from a plant which is stressed in phosphorus. So we apply methods of feature extraction, which are based on what we call frequency domain analysis. Why? Because if you look at this pattern, you can't be able to decipher much from it. And the thing about it is that pattern is not quite replicable. We can't get it across a single class of our plants. So for that reason, we employed the method of wavelet scattering transform to be able to get features out of that plant fluorescent signal. And at the end, we used a classification algorithm to detect whether there is phosphorus or phosphorus is not available in the plant. So we have this season so far. We have 72% accuracy. It's not quite promising, but we are trying to push it further to get more out of it, while trying to do the same thing on a plant simulation model. We have 85% accuracy. So far we have reached, we've gotten an award for our research at the Triple E Conference poster presentation. So we had, our research was voted as the best research by some industry panel, and we've also secured a grant to conduct a pilot in Nigeria, starting from the 1st of June to 30th of, 30th of June, and that is a grant of £7,000 to work together with smallholder farmers. I'll be very happy to have feedback, as well. I'm also open to collaboration, as the whole thing about this research is to drive impact, and the impact is about getting people to have food on their tables and then having a sustainable life on this land. Thank you so much for the opportunity.
So can I ask a question?
Okay.
Sorry, was there someone else who didn't see? So if I understood correctly, so does the pattern of light, so the oscillatory pattern of light you apply, do you get the same oscillatory pattern of expression for like the fluorescence, for example, with kind of like a phase shift? So what was the other thing you were measuring? It was the fluorescence and what were you measuring, the phosphorus?
Yes, when you have a plant which is deficient in phosphorus and another plant which is having enough phosphorus, then you use a source of light, actinic light, at let's say 680 nanometers, and make sure the light, if the light frequency is quite a slow one, let's say a period of 240 or 360 seconds period, so it means it changes quite slowly. So they behave in different ways. Both behavior would have harmonics, and they might also have things like phase shift, but definitely you have harmonics. So you have a sinusoidal input at 360 seconds going in. So the output is also sinusoidal, but with harmonics. So the 360 second signal, which originally came in from the light source, is still present in the fluorescence, but with additional harmonics in it. So what we are doing is to try to see how much information content is available in that harmonics.
Right, so by looking at that, you can predict how much phosphorus is there.
Well,
by
it's
looking at the fluorescence.
Well, we can't predict it quantitatively, but we can, as for now, we can only say whether it's there or it's not there. So that we've been able to achieve it in the first part of our research. And that's where, at the moment, we're having the challenge to see how much of phosphorus is not there or how much is there. So that's the second phase. And at the moment, I think we don't have good result for that.
Right, and have you tried to manipulate the way you administer the light to see how does this affect the pattern, or if it could have any effect on the growth regardless of the phosphorus.
You mean the wavelength?
Yeah, well, the amplitude and the period of the...
Yes, we've been using different periods. So we've done like a sweep. So we started with fast changing signals, and then we moved to slow changing signals. And lastly, although we've tested the last part just on the simulation model, as the device is still under prototyping, so we even tested square waveforms. And the reason why we tested square waveforms is because it's a very rich signal. A square waveform has lots of different signals in it. Let's say you take a square waveform of 240 seconds. So it's not just 240 seconds period. It has other periods like odd multiples of 240 seconds, scaled down to some various values, and then put together. So that has given us even a higher accuracy, which I've not put in this presentation. It's because we don't have it verified in hardware, but we've had it working quite better in the simulation. So of course we do change the pattern. And in fact, when the light is changing quite slowly, you don't see anything. And when it reaches a certain point that it becomes too slow, as if it's a constant light, you also don't see anything. But within that bracket in the middle, when the light starts to change slowly to some level, then that is where we are seeing the pattern. And from the time domain also, we've observed some hysteresis behavior. Even if you go back to the result from here, you would see this is quite hysteresis when you look at it from a perspective of any system's perspective. The light pattern going up is not the same light pattern coming down. So if you take it and normalize the value, you are going to see a hysteresis. And then the hysteresis we've observed changes as the frequency of the light source is increasing, sorry, is decreasing. So the lower the frequency, and then the higher the hysteresis.